Look at a tree standing in a winter forest. Bare branches, no leaves, no movement. Everything about it reads as barely alive. That impression is wrong. Inside that tree, right now, thousands of genes are active. Cells are rebuilding themselves at a molecular level. Ice is being managed, crystal by crystal. The tree is not resting. It's running, one of nature's most sophisticated survival programs. And none of it is visible from the outside. The danger isn't the cold itself. It's what the cold does to water. Every reaction inside a living cell happens in a watery environment. A large tree can be 50% water by mass. And when water freezes, it expands. Most substances shrink when they solidify. Water does the opposite. For a living cell, that expansion is catastrophic. Ice crystals tear through cell membranes and destroy the cell's internal structure. Ice forming outside cells also pulls water out of neighboring cells through the cell wall. The cell slowly dehydrates, losing water to the ice forming right next to it. The tree's water transport has its own problem. Freeze-thaw cycles trap gas bubbles inside the tree's water-carrying tubes. Those bubbles backflow like a kink in a hose. So how does a tree survive this? The answer starts long before winter actually arrives. Trees don't wait for the first frost to begin preparing. They start weeks, sometimes months, in advance. The key trigger isn't temperature. It's day length. As summer ends, the days get shorter. Trees have light-sensing proteins in their leaves that detect how long the days are. Day length is a more reliable signal than temperature. A warm October doesn't mean winter has been canceled. Short days confirm it's coming regardless. The preparation happens in 3 stages. In the first stage, shorter days alone trigger growth to stop and preparation to begin. The second stage starts as mild cold arrives, temperatures between 0 and 10 degrees. Stored starch begins converting into simple sugars. Those sugars become the tree's primary weapon against freezing. The third stage is triggered by the first actual below 0 temperatures. Each stage builds on the previous. The order matters. But dormancy itself is more complex than it sounds. People say trees sleep during winter. That's not what's happening. Dormancy isn't shutdown. It's a carefully regulated survival state. One kind is growth, paused by signals from other parts of the tree itself. The second is an internal lock. The tree physically can't grow yet, even if weather warms. It needs a minimum number of cold hours before that lock releases. This is why a warm winter isn't just inconvenient for a tree, it's dangerous. A tree that doesn't get enough cold hours may never properly wake up in spring. Now look at what deciduous trees do with their leaves. Leaves are the tree's power generators during summer. In winter, they become a problem. They keep losing water through tiny pores, even when the soil is frozen. Leaves also catch snow and ice, adding dangerous weight to the branches. So deciduous trees dismantle their leaves, systematically, and in a specific order. Before any leaf falls, the tree extracts everything valuable from it. Nitrogen, phosphorus, the building blocks of protein, all pulled back into storage. Research has shown trees can reclaim up to 90% of a leaf's nitrogen before it drops. The autumn colors are a side effect of this extraction. The green pigment breaks down first, revealing the yellows and oranges underneath. After the extraction is complete, the leaf detaches cleanly, and the wound seals immediately. Leaf drop isn't a tree dying. It's a tree recycling. Conifers take a completely different approach. They keep their leaves through winter, but they transform those leaves into winterproof structures. Conifer needles have a much smaller surface area than broadleaves, dramatically reducing water loss. A thick waxy coating covers each needle. The tiny pores for gas exchange are tucked into sheltered grooves, protected from wind. And the needles undergo the same chemical transformation as the rest of the tree. Here is where the biology gets genuinely strange. As the tree prepares for winter, it produces its own antifreeze. Those sugars flood into every living cell in the tree's bark and wood. The more dissolved sugar in water, the lower its freeze point, same principle as road salt. So the liquid inside the cells becomes much harder to freeze. Those sugars also cling to proteins, and hold them stable during the stress of freezing. And they keep the cell membrane flexible rather than rigid. One consequence of all this you can actually taste: maple syrup. The sweetness of maple sap reflects the sugar accumulation from the previous autumn. Those sugars were loaded in as protection against winter, and we're tapping them in March. But sugars are only one layer of defense. Trees also produce proteins that specifically interact with ice crystals. These proteins don't stop ice from forming. They act as growth limiters, surrounding crystals and preventing them from getting larger. Small ice crystals are relatively harmless. Large ones pierce membranes and kill cells. The tree concentrates these proteins in the spaces between cells, exactly where ice forms first. Here's the conceptual shift that ties all of this together. The goal isn't always to prevent ice from forming. It's to control where ice forms. Strategy 1. Flood cells with dissolved sugars to lower the freeze point. This works, but only down to a limit. Below -40 degrees Celsius, water freezes regardless of anything else. Trees that rely only on this strategy can't survive the coldest northern climates. Strategy 2. Allow ice to form, but only in the spaces between cells. As that external ice grows, it pulls water out of the neighboring cell. The cell interior concentrates, more sugar, less water, harder to freeze. Eventually, that concentrated interior becomes too thick to crystallize. It solidifies into a glass-like state, rigid, but with no ice crystals. No crystals. No damage. The cell survives. For all of this to work, each cell's membrane also needs to stay functional in the cold. Some of those fat molecules have straight tails that pack tightly together in cold. A membrane full of those straight-tailed fats becomes rigid when cold arrives. A rigid membrane stops working properly. Other fat molecules have a kinked tail, a bend, that prevents tight packing. Those kinked fats keep the membrane flexible, even in cold temperatures. As autumn arrives, trees actively swap more straight-tailed fats for kinktail ones. Cold activates the genes that carry out this molecular redesign. The tree's water-carrying tubes have their own winter vulnerability. When water in those tubes freezes and thaws, gases dissolved in the water escape as bubbles. They form tiny bubbles that can permanently block the tube. Different trees have evolved completely different solutions to this. Conifers have narrow tubes. The bubbles that form are small enough to dissolve back on thawing. Oaks, elms, and ash have wide, efficient water tubes that are highly vulnerable. Their solution: abandon the damaged tubes entirely each winter. Each spring, before the leaves open, they grow a fresh ring of replacement tubes. And then there is the sugar maple. In late winter, the daily freeze-thaw cycle generates positive pressure inside the trunk. When ice melts each morning, compressed gases push sap outward through the wood. That's what drives the maple syrup tap, not spring warmth, but winter's freeze-thaw cycling. Underground, the biology is different. Soil insulates, roots a foot below the surface may be near zero when branches face -20. At 2 to 4 degrees above zero, many trees continue limited root growth and nutrient uptake. Their roots also connect underground to a network of hair-thin fungal threads. Those networks stay active through winter, channeling water and minerals to the roots. The tree keeps sending stored carbon to maintain that network, even while it isn't photosynthesizing. And snow helps. Fresh snow is packed with trapped air, one of the best natural insulators there is. With air at -30 outside, soil under a deep snow blanket can stay near zero. Now for what most people don't know. A researcher named Akira Sakai spent decades freezing tree branches in the laboratory. He submerged fully winter-prepared branches from boreal trees into liquid nitrogen, -196 degrees. He warmed them slowly. The cells resumed normal function. They survived. The tree had drawn every drop of water out of its cells before the freeze. The cell interior had solidified into biological glass. No water left, no ice crystals, no damage. Scientists studying how to preserve human organs for transplant have examined this biology directly. The tree solved the problem of extreme cold preservation long before we knew we needed it. Here's a second finding that surprises researchers. During winter dormancy onset, trees activate more genes than at almost any other time of year. More gene activity going into winter than during much of the growing season. The winter tree is, genetically, running harder than the summer tree. And cold acclimation can be lost. A warm spell in January can cause a tree to start losing its cold protection within days. It loses that protection faster than it can rebuild it. A hard freeze after a mild stretch can damage tissue that survived -20 in December. That tree standing in the snow, you've been calling it dormant. You've been watching one of nature's most sophisticated performances and not seeing it. The biology that keeps it alive doesn't need to be visible to be extraordinary. What's most remarkable is that this transformation happens completely every year and then reverses in spring. See you next time with something just as surprising.